Good evening, everybody. Can everybody hear? Yeah. Give me a... I'm Tina Stevens from the Alliance for Humane Biotechnology, which is one of the member groups of SynBioWatch, which is bringing you the series East Bay Conversations, the Promises and Perils of Biotechnology. Thank you very much for joining us for this conversation on human genetic modification. To facilitate your participation, volunteers, if they haven't come around already, they're going to come around with cards for you to write down your questions and your comments. And to facilitate bringing you more East Bay Conversations, they're also passing around donation envelopes for your tax-deductible donation. No donation is too small, and they really help. Thank you in advance for that. And our thanks also to the CS Fund, whose generosity has seeded this event and helped us get our beginnings. Now, when you came in, you received a fact sheet and a letter concerning human egg harvesting, and I'd like you to hang on to that. Later on in the evening, we'll let you know why it's relevant to some of the things that we're talking about tonight. Our host this evening is Milton Reynolds. Milton is a senior program associate with Facing History and Ourselves, using original publications and teaching guides on the Holocaust, on other genocides, on mass violence. Facing History works with educators to enhance student civic learning by identifying the sources of bigotry and prejudice. Milton's area of specialization in the American eugenics movement and his incisive insights on how historic legacies manifest themselves in our society today makes him a very fitting host for tonight's consideration of human genetic modification and what has come to be known as techno-eugenics. East Bay Conversations is very happy that Milton agreed to do this. Thank you, Milton, and thank you for coming.
Thank you, Tina. Great to be here. And I also extend a welcome to everybody. It's nice to see the audience growing. We had a great exchange with the first iteration. I'm looking forward to another good one tonight. So tonight I'll be in conversation with Stuart A. Newman. Stuart is a professor of cell biology and anatomy at the New York Medical College in Valhalla, New York. His early scientific training was in chemistry, but he moved into developmental biology, both experimental and theoretical, after receiving his academic degrees, naively anticipating that he would be working in a field of pure science with no commercial or military application. Stuart has contributed to several scientific fields, including biophysical chemistry, embryonic morphogenesis, and evolutionary theory. His proposed mechanisms for the Cambrian explosion at the origin of the animals, the patterning of vertebrate limb skeleton, and the origin of birds are under active investigation. Stuart has clearly been busy. Newman was a founding member of the Council for Responsible Genetics in Cambridge, Mass., and is currently a member of the advisory board of the Alliance for Humane Biotechnology out of Berkeley, California. He has been a consultant with the National Institutes of Health on policy regarding the use of human fetal tissue, and for research has testified before congressional committees on issues ranging from the patenting of organisms to human stem cells and cloning. He has also been an interim fellow at the Pasteur Institute in Paris, a Fogarty Senior International Fellow at Monash University, Australia, a visiting scientist at the University of Paris Sud, the French Atomic Energy Center at Saclay, and the Indian Institute of Science, Bangalore, as well as the University of Tokyo. He is an external faculty member of the Konrad Lorenz Institute, Klosterneuburg, Austria. In response to the growth and patenting of life forms and the emergence of technologies that threaten to blur the boundary between human and non-human organisms, Stuart applied in 1997 for a patent on a human-chimp chimera, which is a mixed species organism, as a challenge to the existing patent policy and a way to prevent inappropriate uses of the technology. He is co-editor with Gerd B. Mueller of Origination of Organismal Form, Beyond the Gene in Developmental and Evolutionary Biology, MIT Press, 2003, and co-author with Gabor Forgasis, I hope I didn't mangle that, of the textbook Biological Physics of the Developing Embryo, Cambridge University Press, 2005. Stuart received an A.B. from Columbia University and a PhD in chemical physics from the University of Chicago. So, thank you, Stuart. Appreciate being here with you tonight. So, let's get the conversation started. I'm struck by the idea in your bio that you suggested that you wanted to enter the field of science, and you did so naively, thinking that you would not be pulled into applications of science that have either a military or commercial application. So I'm wondering, how did your bubble get burst, and what did you do as a result of that? I'd love to sort of maybe look at the trajectory of your career as a way of situating our conversation.
Right. So I was a graduate student at the University of Chicago during the 1960s, and Chicago was one of the main sites of the Manhattan Project, which led to the development of the atomic bomb. And I was a chemistry student, and it was always kind of hovering over people in chemistry and physical sciences that the science that we were doing was not simply for its own sake, but had applications, and some of these applications were pretty horrendous. So that had happened in the 40s, the development of the bomb. In the 60s, the field of chemistry was developing incendiary weapons and napalm and things that were being used in Vietnam, which was the war was going on while I was a student. And again, there was this consciousness of how our scientific efforts were not pure efforts, but were being applied. I became interested in the new areas of developmental biology during this period, and it seemed to me that it was unquestionable that nobody would ever conceive of using developmental biology for any commercial purposes or for reconfiguring human life. It was the science was very primitive at the time. It was very exciting because there was so many unknowns, and the complexity was so great. How do embryos develop? It was a relatively obscure field. There were not too many people working in this area, and the lineage of the field went back to Aristotle, who observed chicken embryo development. So it was philosophical, and it was quite mysterious. While I was a student, as I finished up in 1970, there was a conference at the University of Chicago commemorating an event that was associated with the development of the Hiroshima bomb. It was the 25th anniversary of something called the James Franck letter and the James Franck report. James Franck report was a report that was put together by a number of members and former members of the Manhattan Project, some of whom worked at the University of Chicago. James Franck, a Nobel Prize-winning physicist, was among them. And this group, in 1945, in April of 1945, warned the president, who at the time had just become Truman, not to use the atomic bomb on population centers. They had said that, first of all, this would lead to a tremendous devastation of human life, and also would create a situation, and they had a lot of foresight, would create a situation in the future where there would be competition around atomic weapons, there would be an arms race, and the United States, were to use this weapon, would lose international credibility in being a force to prevent the spread of nuclear weapons. And this, of course, is exactly what happened. And we're living in the world that is the legacy of the denial of the recommendations in the Franck report. So the University of Chicago in 1970 had this commemorative meeting of the 25th anniversary of the Franck report, and this was the first I had heard about this. And most people don't know about it. If you poll the vast majority of scientists, and certainly the general public, knows nothing about this. But these were some of the premier scientists of the time. Glenn Seaborg of University of California, Berkeley was among the signatories of the Frank letter that arose from the Frank report. This is a landmark in the history of the kind of scrutiny of science by scientists. It was really a first of its kind, and it really, I think, should be part of the education of every scientist, but of course it's not. Well, as I got into my own field of research, which became developmental biology, in the 1970s there was a great flourish of technological breakthroughs that allowed the sequencing of genes, the manipulation of genes, the insertion of new genes into organisms, creating new kinds of organisms, or at least new versions of organisms. And this immediately became very commercialized. Scientists who might have considered it taboo to have commercial connections and commercial money took to it very readily. And this is the culture that we're living in now. It's very commercialized, and the justification of a lot of this work is how are we going to remake humanity? It's not, since it's still distant, given the existing technology, people are not proposing to do it tomorrow, but this has become the ideal of the field, and it's certainly the basis on which people raise money both commercially and governmentally.
Great. So there's a couple of themes there I'd love to pick up on as we sort of move through the conversation. One is this issue of historical understanding, and how do people's understanding of the past, how does that shape the way in which they show up in the present? And the other one is the sort of the growing sort of, and the different incentives and what I might think about of as eugenics in a more traditional sense. But I know in the promotional materials there was some mention of your 1997 patent for the humanity, which you remind me is a chimera. But I'm curious what happened. And you issued this patent, and so first of all, what happened with the patent? Was it accepted? And more importantly, what was the conversation that I believe you were trying to provoke by in that act, and how does that conversation go?
Right. So among the technologies that had come about in this kind of new flush of developmental biology research that followed the ability to manipulate genes, to manipulate embryos, was the production of chimeras, which are mixtures of the embryo cells of two different species. And these mixtures can develop into coherent organisms, very surprisingly. So I'll just jump ahead to the motivation of the patent application, and then go back to this technology.
Great.
So I was approached in the mid-1990s by the economist and social critic Jeremy Rifkin, who put a challenge to me. He said, is there anything you can think of that is scientifically feasible, that can actually be done with existing technology, that would be very useful, but would be so beyond the pale, so outrageous that it would raise questions about how technology, even if well motivated, can be go too far and used for very disturbing purposes. So I thought back to this experiment in the middle 1980s of the sheep-goat chimera, which the investigators who did the experiment called a GEEP. And these were agricultural scientists, and they were interested in not so much the pure developmental question, but they were interested in the ability of hybridization. Different plants, of course, can be hybridized. Animals are much more difficult to hybridize. They will not breed true and so on. But what they did was they took the embryo cells of sheep sheep embryos and goat embryos and mixed the cells together. And they found that the cells, even though there was more than 12 million years separating these lineages in evolution, the cells could recognize each other as embryonic cells, and they could cooperate to make an attacked organism. And the organism that resulted was midway between a sheep and a goat. And if the cells in this animal were tested, it was found that every cell was either a sheep cell or a goat cell. Nonetheless, they took appropriate places in the animal's tissue and made livers and kidneys and brains and so on. It was a kind of a normal seeming organism, but nothing that had ever existed before. And there was some scientific implications of this. They tried grafts from the different species to this new organisms. They tried grafts from that organism to the parental species and so on. And they found out some important issues about the immune system and how it develops. So I said to Jeremy Rifkin, if we took this technique and applied it to a human-animal mixture, that is, use human cells, human embryo cells in a chimera, either with mice or with pigs or with chimpanzees, we would get an organism that was part human and part non-human. Now, what would be the motivation and the practicality? Because if you applied for a patent, which is what I was suggesting to do, you had to show that it was useful, it was beneficial. We don't grant patents to things that are not beneficial. So what would be the benefit of this organism? Well, for one thing, it could be used as a source of transplantable organs. Because of the immune tolerance in these chimeras, you could actually expect that the heart of a chimera would be tolerated by a human recipient, or much more than the heart of a chimpanzee, for example, or a gorilla. So it could be the source of transplantable tissues. It could be they could be models for response of human tissues to new drugs. Sometimes a drug is developed and the permission to use it on human subjects to see if it has a desired effect is very difficult to obtain. But these organisms would have no civil rights. They would just be our creatures to do what we will with them, and so we could use them. So basically, this was a Swiftian modest proposal. It was making these part human organisms and proposing all sorts of uses that they could be put to. And so with funding from Jeremy Rifkin's funding sources, which I think included the CS fund, by the way, I applied for this patent. The legal costs were very high. The amount of work was very extensive, but it got a rise out of the patent office. The patent office hated it. They invoked initially a 1924 provision that said that they won't patent anything that violates social norms or is offensive to the public. And they had to go back that far to find that precedent. But more technically, they questioned, they said that this was inappropriate subject matter because in the way it was initially described, it was difficult to determine how human this organism would be. So I wrote a response that said, okay, I'll just control the number of cells. I'll put in very fewer human cells than chimpanzee cells, so the outcome will be less than half human. Is that good enough? And then they kind of changed the subject and they found some other technical things that they didn't like about it. And it went on with cycles of revision until about 1996, I think. Well, it was about a 7-year process. 2006, 2005, 2006. So it was Yeah, and we just kind of threw in the towel. It was getting very expensive, but it also had the desired effect. There were at least 50 law review articles that mentioned this proposed patent as raising constitutional issues. And there were television documentaries, there was an article in Mother Jones, there were, it really kind of percolated through the culture, and people knew about this issue. It was discussed in Congress, and it basically raised the level of awareness. And at the time that the patent was applied for, and it was disclosed when the first rejection came through in 1998, the person who had devised something called the Harvard mouse, which was a mouse that got cancer at very high rates, unexpectedly, but then the investigators saw a benefit that it could be used for cancer research. But anyway, the person who devised that mouse went on television and said that this chimera issue is a total scare tactic, because no scientist in their right mind would ever make an organism that was a chimera between a human and a non-human organism. And then just a couple of years later, people started doing it in the laboratory. They couldn't patent it because we had already disclosed the idea, but they were doing it, and they weren't doing it to make full-fledged part human organisms, but they were looking at the communication of the cells at the early stages. So, of course, anything that's possible to do will be done. So that's the lesson.
So that's great. Lots of pithy things there to pick up on. I guess one is, we're 16 years removed from the issuing of your patent, and I'm wondering how the topography of the landscape has changed, both in terms of technological capacities, but also the marketing. I know that oftentimes in sort of market-driven economies, you begin to hype things long before they become a reality, and so I'm actually curious about this. Is there a leading edge of marketing for eventual economic opportunities, but also the technology? How has the technology changed? I'd love to touch on both of those.
I just want to qualify a point. So the patent was never issued in the sense of being awarded as a patent. It was applied for, and it was turned down and reconsidered, but it was eventually denied rather than issued. But the science has moved on, and there are a number of different technologies that have been proposed to be used on people. So why would people propose using it on humans? If I can, I'll go back to the early part of the 20th century, and when genetics first became established as a science, and Mendel did his work on the genetics of plants, peas, and other plants in the middle of the 19th century, but his work didn't become really incorporated into the scientific mainstream until 1900. But in the early 20th century, genetics began to take off, and the geneticists of the time raised the prospect of using this science to improve the human species. And so they actually had the ear of a lot of legislators. They said, if we have people that probably have bad genes, we should keep them from reproducing. So actually in California, there was a very draconian law, as there were throughout the country, but California, more people were involuntarily sterilized in the name of eugenics than in any other state of the Union. And this continued through in the United States, in Europe. Actually, eugenics was what was basically a kind of concept of Francis Galton, cousin of Charles Darwin, working in late 19th century England. And there was a very active kind of eugenics movement in both the United States and England, and then throughout Europe in the early part of the 20th century. And this was not just an idea of conservative or regressive people. Some of the most progressive scientists of the time advocated the use of genetics to purify the human species. And very famous geneticist, one of the great quantitative geneticists, J.B.S. Haldane, who was a Marxist and a very progressive person, he wrote a book called Daedalus, which advocated using genetic ideas and in fact anticipated being able to modify genes, genetic engineering, to create superior people. And one of Haldane's childhood friends was Aldous Huxley, who saw the negative side of this and wrote the book Brave New World. That kind of came out of these eugenic dreams of his friend Haldane. In any case, in the United States, one of the cutting-edge institutions for genetic research, as it still is, was Cold Spring Harbor in Long Island in New York, the Cold Spring Harbor Laboratory. And the head of the Cold Spring Harbor Laboratory was a guy named Charles Davenport, who had been a professor of genetics and biology at Harvard. He took on the directorship of the Cold Spring Harbor Laboratory, and he kept a repository of eugenic data, or at least reportedly eugenic data. He did studies of people in the island of Jamaica, and documented what he considered the deterioration of humanity as interbreeding between black and white people took place. There were critics of these eugenic ideas at the time, but the critics did not hold sway. Davenport was able to persuade the United States government to impose laws restricting immigration from certain countries. There was an immigration law of 1924 that Davenport was important in formulating. And Davenport had a very active scientific, pseudo-scientific, let's say, interaction with German scientists who were at the leading edge of the coming Nazi ideology of eugenics. And it was really the implementation of eugenics during the Nazi period that turned a lot of scientists away from at least overtly referring to eugenic motivations in their work. But it remained there. In fact, a later director of the Cold Spring Harbor Laboratory, another former Harvard professor, James Watson, was a very avid eugenicist and made no bones about it. The co-discoverer of the structure of DNA. And these ideas continue. But as new technologies became available, as, for example, more genes became identified with disease conditions, the idea of the genes as kind of the secret to life and the blueprint of organisms, the program of organisms, led to proposals that once we crack the code of the blueprint of human life, we'll be able to reconfigure it and make better people. And every time some new advance occurred technically, there were people associated with those new advances, Watson being one of the prime examples. These people got the ear of government officials, they got the press's attention, and they were kind of given a kind of leave to spout off on what they saw as the brave new world in front of us. And this happened with the discovery of DNA. It happened with the potential to modify the DNA of organisms, which came in the 1970s. The genetic engineers of the time You can look at the press that surrounded these discoveries. People made claims about the genetic future for humanity. When cloning was done in the 1990s, the chief technology officer of Microsoft at the time, Nathan Myhrvold, wrote an article extolling the great future of cloning humans, and Senator Tom Harkin, the same, said he looked forward to the time when we would clone people routinely. Then with the newest flush of this technology, synthetic biology, there are many examples of articles in the popular press, the New Yorker, the New York Times, so on, where principal people in this field are talking about the prospects of making better people with these technologies. So it really doesn't end. It's the science moves on, and the ideology remains with us.
I think that's a really important point. I mean, I guess we've spoken about this a little bit in the past, and I've worked for this organization facing history ourselves, and one of the sort of advantages of working with an organization that actually works on history is I get to sort of wallow in history, quite frankly, on a regular basis. And sort of two of the resources I work with are this one, Race and Membership in American History, the eugenics movement, and the other one is Holocaust and Human Behavior. And I think that when Alan Stoskoff, who was the primary writer of these texts, sort of initially conceived of it, they were sort of companion pieces that would speak to each other. And, I'm invested in living in a just society, and I've been intrigued by the history. And sort of my reading has taken me into other societies. And what I've come to sort of recognize is that, to a certain degree, race is sort of a central organizing principle of modern society. And so we see the ways in which societies use policies of immigration, healthcare, education, and a variety of marriage restriction, a variety of other things as a way of maintaining the health of the nation. And so if the nation is conceived as a racial body and institutions are purposed towards that goal, then even if there's a confrontation with a set of ideas, it doesn't deal with the intellectual inertia behind those ideas. And so one of the things I'm struck by, quite frankly I was struck by when I first read the text, is that I had no real knowledge of eugenics. My middle name is Eugene, so I had some sort of I'm not sure what that was all about, but as I moved into the history, I was struck by how something so central has been hidden in plain sight. And so, I'm wondering, if you think about the rise of these technologies and an absence of historical knowledge, can you imagine that there's a way in which, if the history was more known to people, do you think that would serve as some sort of mediating factor, or do you think that the drivers behind these technologies and the incentives associated with that are too great to sort of step back and take another look?
Well, there are very strong forces trying to obfuscate the science and misinterpret it and apply it to kind of perennial prejudices. It's one thing that we're seeing now is that the analysis of the genome is occurring on a very fine level. So there are clear differences between every human individual in the versions of the genes that they hold. I mean, we all have the same set of genes, but we all have variants of those genes. And some of the variants are associated with some groups of people and other variants with other groups of people. So given the history of racism and prejudice in the country, you can imagine that people might map those differences to their prejudices about characteristics of the groups. Well, what the truth about these genetic differences are is that the closer geographically any people's ancestors were, the more similar their genes will be. So people from the same geographical region will tend to have similar genetic markers. Often these are associated with ethnicities or with physical appearances. But if you try to look at issues that people in kind of the vernacular prejudices associate with one group or another, certain physical characteristics, skin color, and so on. These are distributed geographically across the world in a way that doesn't correspond necessarily to the genes. So some people that look alike to the common view are as more genetically different than people who look different. So there's not a good mapping there. The mapping is a geographical mapping, and but the mapping of people's ideology, the mapping of prejudices about ethnic groups and so-called racial groups is different. And so people are constantly going to try to bring their map of prejudice to the map of what's found about the genome. But it's less and less tenable to be able to do that. But that doesn't deny that there are genetic differences that are geographically associated, and therefore, and because of these genetic differences, you may have groups of people that react to medications differently than other groups of people, and so on and so forth. But it does not correspond to people's notion about what races are.
So one example that I think, and correct me if I'm wrong, is something like sickle cell anemia. People oftentimes think about that as a race-based trait, but in fact, as I understand it, it's an environmental adaptation to the presence of malaria. So the body's sort of ability to sort of to cope with that, and sort of within that mind, it occurs to me that the certain amount of genetic diversity is actually is necessary and healthy. And as I think about a lot of these new technologies, it seems like there's at least maybe from my perspective, there's a tendency to narrow down to traits or things that are believed to be or have historically been defined as ideal. And I guess from a layperson's perspective, which I would consider myself, I see great danger in that. Is that something that resonates with you, or is that something that is way out of left field?
Well, this question of the ideal, of like what are the ideal traits, it's really a power-based thing. It's every technology, every science. There are parts of the science that are objective and represent progress over previous ways of understanding things, and there are parts of the science that are just misinterpretations, and the misinterpretations or the ambiguities, because when we find something new in science, we don't always know what it means. We know that it represents something different than what we might have expected. It represents a partial explanation of something, but we don't totally know what it means. And then the people who can exploit those ambiguities are the people with access to the media, to wealth, to funding, and so on. So any valid science and any ambiguous science is going to tend to be used by groups that have power to enhance their power, often against other people with less power. So it's always something to look out for. And the idea that we humans are doing science and we're all marching forward to the future in the name of all of us. This is not the way things work. Some people do science, other people pay for the science. The people who do science have to cater to the people who are paying for it, and often catering to prejudices in addition to catering to real needs.
So I think those are all really important factors to sort of take into account. And I'm also thinking about these unintended consequences. I think science, as I understand it, can sometimes be messy. You go up with a certain hypothesis, a certain idea, you believe some things are going to happen, and it doesn't happen the way that you would hope it would. And certainly my read on eugenics, sort of accepting the fact that there were some people who were pretty rank and racist in quote-unquote a classic sense, but there are also individuals who really did have this utopian vision of the future. Why wouldn't you try to create a healthier society? And in many cases, they had no idea or couldn't comprehend what might happen with those ideas. And so if we think about, the dynamics of human behavior, in response to technologies. Where's this path leading us? I'm imagining some of the things that people are proposing now may have some pretty horrific unintended consequences that we may not be able to contemplate at this point. How do you?
So classically, when we look at eugenics, at the use of whatever information we might have had at any period in history to improve humans. So classically, we've had negative eugenics, where people who were considered genetically unfit were discouraged from reproducing and contributing to the gene pool, and the sterilization, the forced sterilizations of the previous century were just a classic example of this. And of course the Holocaust in Germany, prime example. Then there was positive eugenics, also used by the Nazis to encourage what they considered desirable to reproduce, the Aryans in that case. When we get to modern technological eugenics or techno-eugenics, technically driven biological modifications where it's possible in principle to modify the genes of developing embryos and to attempt to make them come out differently from the way that they would have otherwise. Positive and negative eugenics take on a different cast, because you're no longer dealing, as you are with the previous era's eugenics, with at least outcomes that are based on kind of evolutionary sound starting materials. If you encourage people to reproduce, they're reproducing typical human embryos of some sort. If you discourage people from reproducing, you're preventing the development of those embryos. If you do prenatal selection of embryos that are considered desirable relative to ones that are not desirable, at least the ones you're getting are kind of naturally produced embryos. They are made of the ingredients that evolution has come up with to make a human being. When you start to manipulate embryos, adding genes, adding parts of cells, cloning, adding mitochondria from other individuals, as being proposed in Britain now, and actually looks like it's coming close to being approved. This is the idea that because the mitochondria, the organelles that extract energy from fuels in the cells, in the egg cells of some women, are mutated, their offspring may sustain certain quite impairing diseases. So to prevent that from happening, the idea is to take the nuclear genes, the genes of the cell nucleus of the woman who is basically going to be the prime maternal agent in this process, put her nucleus in the egg of another woman whose own cell nucleus has been removed, and then fertilize that mixed egg, that is fabricated from two different cells with a sperm. Then you get an individual with three parents, and probably or hopefully free of the mitochondrial condition. But when you're doing this, you're really taking damaged cells and putting them together and producing a human being not from the kinds of ingredients that evolution has come up with to make a person, but from parts of these cells. So you're doing an experiment. You're doing something where you may or may not get the outcome you desire. And if you don't get the outcome you desire, what do you do with the result? Do you discard it? Do you say, well, that's what happened? I mean, people who want to do this are not willing to say that that's what happened. They're willing. They want to go for something better. So you're really kind of moving into a world where we think of our offspring not as fellow members of our species who are owed respect, but as kind of products of our technology that could be made better. And if they're not, well, try again.
So it occurs to me, I think, if I get this right, you're referring to, was it pronuclear transfer and maternal spindle transfer, where you're literally sort of recreating different human species. I suspect that there's a variety of reasons why that might be problematic. One is, what do you do with people who may not be, quote unquote, ideal in terms of what you're trying to create? But don't we, by virtue of sort of proposing these ideas, step into a new realm where there's a focus on perfection, which may not only create false hopes, but also cause us to reflect on the human condition in ways that are even more profoundly problematic. And we started to have this exchange a little bit earlier, and maybe it's a little bit off topic, but I was telling you that I recently got married about 2 years ago, and sort of being older, getting married, and my wife's a little bit older, we entered into this union with this idea that we might have kids, but that there might also be just a reality that we may have waited too long, and that is, to me, a reasonable calculus to make, and that's an acceptable outcome. So should we try to usurp the trajectory of human evolution and intervene when we can on the sake of science? I mean, how do we reconcile these challenges?
Right. Well, people are motivated to, in many cases, to have children and to go through what it takes to do it. I think that there's a rational place to draw a line in this. I think that there are all sorts of assisted methods that people, and some of them are not without problems, when a woman is stimulated hormonally to produce a sufficient number of eggs to do in vitro fertilization. But at least some of these procedures impair her health in producing these eggs. But nonetheless, when a child is produced in this way, you're using an egg and you're using a sperm, and what you get is an evolutionary product called a human child. If you start to resort to taking bits and pieces of cells and putting them together because you want to go to extremes, I think that's the place to draw the line. I think that the manipulation of a human reproduction in such a way that causes you to use non-standard ingredients is really an experimental enterprise where many times it's just not going to work. I mean, anybody who's done biological experiments knows that at the beginning it mostly doesn't work, and then you find the conditions that make it work, and then if you can get it to work 60 or 70% of the time and it's statistically significant, then you publish it, it goes into the scientific literature, and people consider that that's the truth, and it happens that way. But it's always variable. And the question is, do we want to produce human beings in an experimental way where it's going to take a long time before the conditions are optimized, and even when they're optimized, they're not going to be ideal? So that's really a line that I think many people have written that it's just not really not rational to cross that line.
Yeah. It seems like moving from an idea of trying to prevent or to cure illnesses, we're trying to do something fundamentally different.
Trying to fabricate prospective people. There's one thing about curing the illnesses of an existing person, and that's kind of a noble activity. Then there's the prenatal intervention where you might say, well, I have these 6 embryos and maybe 2 of them look healthier than the other 4. Well, again, you're getting kind of naturally produced offspring, but you're doing selection. So there's a eugenics aspect to that kind of selection. But then when you start to actually manipulate and create novel forms with the hope that they're going to be better human beings for it. I think that's a good place to draw the line.
I would tend to agree. So we're getting probably pretty close to time to opening it up for some questions. Let me throw out one more. I mean, I think, again, and sort of thinking about this historically, I'm always sort of thinking about, again, the unintended consequences, and in many ways, sort of the disparate impact on particular communities. I know when I look back on the history of eugenics, I know that, in many ways, women bear the brunt of a lot of reproductive surveillance, or the disability community is targeted, and sort of tropes of deficiency or decay are many times are voiced upon them as a way of sort of marketing perfection, whatever that means. I'm also thinking about the consequences for the poor and the various different communities who might be affected differently. As you see the inertia of these technologies moving forward, it also raises the question of their need to source more gametes, right? And I know it's probably easier to source from sperm than it is to source eggs. So as we think about the social justice implications of these technologies, would you be willing to venture sort of a guess as to some of the consequences that we might see, or is it too early to?
Well, I think we're already seeing that. I mean, the outsourcing of reproduction, people from affluent societies going to less affluent societies and hiring surrogate mothers of gestators of their embryos. The question of eggs, the extraction of eggs, it's for a woman who is trying to have her own child, she might accept the impact, and there might be social pressures on her to accept too great an impact on her health from doing these procedures, but at least she has a choice of doing that. When it comes to people who are very badly off, and there's a question of needing eggs for either other people's reproduction or even now research. I think that there's a move in the California legislature to approve the solicitation of eggs for research purposes, not even reproductive purposes. Certainly people who are worse off, more desperate, are going to allow themselves to be subjected to that. Maybe they don't have other options. So there's this whole question of people's kind of reproductive potential being commodified.
So just for my clarification, so it sounds like we're moving in a direction that is different from the gamete sort of ads I used to see in, so whether the Stanford rag or the Cal rag, where you're looking for people with certain phenotypic qualities, high SAT scores, Ivy League credentialing, those sorts of things. So it sounds like the market for eggs has grown, and we're looking for a broader spectrum of people, often through the harvesting, for lack of a better word.
Right. Well, if it's for experimental purposes and not for reproductive purposes, you can go deeper into the population.
Great. So I know that Tina's been circulating, and there's a variety of questions that have come from the audience, and so I think it'd be great to spend a little bit of time getting a sense of what folks are thinking about. And let me stop peppering you here for a little bit. So here's the first one. Let's say I understand where you were drawing the line, but how can we prevent people from crossing the line? Is it legislation? Is it moral persuasion? I mean, how do we create safeguards, or can we?
Yeah, this is a very complicated question, particularly in the United States. Legislation in the United States around anything to do with embryos is very difficult because it's so contentious because of the divisions on the abortion question. So anything that comes to Congress that talks about not doing this or that to embryos smacks to people who are trying to protect the right to abortion. It smacks of protecting the embryos, so they won't let it go through, even though it might be rational to let it go through. In Europe, there's more of a at least not in England. In Germany, in France, in Italy, there's more of a kind of willingness to legislate against these things. Germany, because of its history, there's a very strong kind of cultural prohibition against techno-eugenics in Germany. In France and Italy, partly because of the religious history. But in the United States, the so-called compromise, or the alleged compromise that's been reached, is that you can't do anything on human embryos with federal funding, but if you don't take federal funding, you can do almost anything you want. So in the United States, there are no regulations against manipulating human embryos, and the idea of passing laws against it seem almost a fantasy. So I think in the United States, the most important thing we can do is talk about the desirability of not doing it and kind of create some kind of social disincentives and public taboos against doing these things.
Seems like in the context of sort of free market capitalism, the lack of sort of regulation presents a very different piece than we've had to grapple with in the past.
This is and I'm glad you bring this up, because people are familiar with the book Brave New World, and it kind of in contemplating this techno-eugenics future. If you speak to people currently who are proposing the same sort of thing, they'll say, "Oh, this is nothing like Brave New World. Brave New World was the government doing it. This is just people by their kind of consumer choices deciding to do this, and that's going to be a benign thing, because people should have the right to make any consumer choices they want." So basically, this whole ideology of the market has kind of moved in to almost cleanse the notion of eugenics of its horror.
So here's something that connects back to some of the things we were in conversation about earlier. So do you think the denial of your patent application set a lasting precedent? And what's likely to happen next in terms of patenting human-animal life forms?
Well, one of the things that the chimera patent application actually accomplished is that because a patent, not only does it have to be useful and have to be feasible, it has to be inventive or novel. Anybody who comes forward now and says, "I'd like to patent the human-animal chimera," won't be able to do it because it's already been disclosed. The idea is out there. So that's been one effect that it's had. But just because something isn't patentable doesn't prevent people from doing it. And as I mentioned, research labs have been doing it. A lab reported recently making rats with whose brain cells are human, and actually human brain cells are quite complex compared to rodent brain cells, and these rats actually run through mazes much faster than non-chimeric animals. But so the patent proposal has had an effect. It's also it stimulated this literature in the legal scholarship about the legal status of chimeras and the violation of constitutional provisions and so on. So there is some effect that it's had that's lasting, but I can't say that it's going to prevent these things from happening.
Really makes me rethink going through the corn maze around the holidays and how to sort of improve my time. Let's see, so here's another great question. So if the U.S. doesn't undertake this sort of research, what's to prevent other countries from doing it anyways?
Well, as I mentioned, many other countries have more prohibitions than the United States. So there are things in these other countries that will prevent them from doing it. And I don't think everything should be looked at as a kind of economic competition. If it's wrong to do, and other countries do it, that doesn't justify the United States doing it.
Great, great. Let's see. So is there really a way to curtail eugenic outcomes that are the result of individual reproductive choices?
Well, not entirely. And people who defend techno-eugenics will say that just our choice of mates is eugenics, and things like genetic counseling, as Troy Duster, who was actually here this evening, has written. Genetic counseling is very clearly a form of eugenics. Eugenics is really very pervasive, and then the question becomes, what kind of eugenics? Where do we draw the line? And it's becoming more difficult to exclude a lot of assisted reproduction, which is also eugenics. Pre-implantation selection is eugenics. It's difficult to prohibit something like that in a blanket fashion. But one thing that we can argue very strongly against is to do interventive eugenics, to genetically engineer or clone human beings. The genetic contributions to these common conditions, and it turns out that there's just a whole network of interactions, and the same condition in one person may be associated with one gene variant, and in another person the very same condition is associated with a different gene variant, and in some cases it's the cooperation between genes and the strengths of their interactions rather than their actual variants. So it's very difficult to discern, and therefore you might be able to survey thousands of genes and stick them into a computer and come up with some set of probabilities, but it would be very difficult to make a decision on whether to carry an embryo to term based on these uncertainties.
So here's a question that I think is a great follow-up to that. It states that if most human traits aren't based on the expression of a single gene, will designer babies really even be feasible? And so I think we're looking at sort of this intersection between the sort of the hyping or the marketing and the technological possibilities. So,
yeah. I mean, I don't believe that they will be, but it doesn't prevent people from trying. In China now, they're trying to discern the genes that contribute to high IQ, and I don't know whether the idea is to select embryos that have those genes or to put those genes in embryos, but certainly people are never ceasing on moving in this direction.
Okay, so we've got a question about some particular patents. So we're talking about the BRCA1 and BRCA2 breast cancer genes. And the question is, what other genes have been patented? And I guess it sort of is related to that. What are the implications of patenting genetic material?
Yeah, there have been a whole number of genes that are associated with rare diseases that have been patented. It's interesting that except for BRCA1 and BRCA2, there are just about no genes that have that kind of impact on the probability of cancer. So even cancer is its genetic underpinnings are uncertain. And the Supreme Court is currently deciding, in the midst of deciding whether the patents held by the Myriad Corporation on BRCA1 and BRCA2 will be sustained. These are genes, they're gene variants. So everybody has these BRCA genes, and they're involved in DNA repair and replication. Everybody has them, but not everybody has the same variants, and some variants are associated with high propensity to get breast and uterine cancer, breast and ovarian cancer, and the diagnosis for these genes is based on the sequence of the gene, and that information with that use is patented by the Myriad Corporation, and it's really it's scandalous because the cost of the diagnosis is very high. It's hundreds of thousands of dollars, and people who might have their lives saved by having these tests can't do it because of the cost.
So it seems like there's a related question, somewhat related. It says, what applications of synthetic biology do you expect to see in the next, let's say, roughly 10 years? And are dangers or benefits more likely? And again, this is sort of this cost-benefits analysis. Where do you think we're headed?
Well, synthetic biology, it's kind of a hyped-up form of genetic engineering, it's generally it's the engineering of genes in concert with each other. So it's not a single gene that is modified, but a circuit is produced, some kind of network of interacting gene products that do a particular task. And there have been proposals, and I think they seem feasible, and I think they work, of having plants that are sentinels for toxins, so that the plant will turn a certain color if it's exposed to a toxin. So you could plant these, you could plant these organisms, and they might turn a certain color if pollution gets high enough. So this kind of thing is probably in the cards. There've been promises to use synthetic biology to genetically engineer people. I think that's it's really not on the table.
Okay, great. And then we're probably getting close to 9.
Quarter till.
So we got time for a few more questions, and then. Okay, so let's get to a few more of these. So can you talk about the human biome? What is it? And whether engineering or modifying the microbiome is a form of genetic modification?
Well, the microbiome is the population of bacteria that lives in the human gut. And there are very many of these bacteria by number, and there are many species of bacteria. And more and more it's becoming associated with person's physical state and health status. We're just at the beginning of understanding this, but it turns out that people, like children that are diagnosed as autistic, for example, will tend to have different populations of bacteria in their gut than other children. And the question is does the microbiome, this population of bacteria, contribute to the symptoms of autism, or is there something biologically changed or physiologically changed in these children that make them susceptible to harboring different populations of bacteria? And most of this is not known, although some people's health have actually been improved by transplanting fecal material from a person with a different microbiome. So they're actually beginning to intervene and bring about changes in health status by manipulating the microbiome. I think that this is a kind of an interesting area of medicine. It's not like genetic engineering in the sense that you're looking at already existing people. You're not changing their development. You're changing their physiological state by altering the organisms that reside within them. But I think it might actually be a kind of rational approach to it kind of acknowledges that humans are really interacting with their environment. They're interacting with the environment of organisms that they're exposed to, and it represents a kind of a scientific advance rather than simply a manipulative technology.
Okay, great. So here's actually a related question. It says, how does the new awareness of the biome as part of the human in quotes, body and the definition of corporations as persons change the concept of humans? And does that blindside our ancient narratives of prejudice?
I think that's something everybody should think about.
Well stated. Can you explain why women's eggs are necessary? And how do researchers get these eggs?
Okay, well, I think it's meant to, why are they necessary for research?
For research, yeah.
One simple answer is that with the increasing, with the rise of assisted reproduction, many women are deferring having children till later in their lives, and since the eggs tend to sustain damage that is in proportion to the age of the woman, a woman might want to harvest eggs when she's younger and freeze them away and use them later on in her life when she's ready to have a child. So one research area is called vitrification. It's if you just take human eggs or mammalian eggs and stick them in the freezer, they won't do very well. Ice crystals will form in them and so on, and they'll get damaged. So you need to put fluids that penetrate the egg and protect the internal structure from becoming damaged by the freezing process, and that's called vitrification. And but we don't know exactly what the best method is, and we've experimented with mouse eggs, and we have good methods for mouse eggs. We have some reasonable methods for humans based on the mouse studies, but some people feel that they would make more progress if they actually had human eggs to experiment with. So that's one thing. Another thing is people would like to explore the development of human disease at the embryonic state. So let's say there are people who have cystic fibrosis, there are people who have muscular dystrophy. We know what they're like as children, as adults. How does the disease develop in the embryonic state? How is it manifested as the embryo develops? Well, we can't know prospectively unless we start with embryos with a known genome. So the idea is if you take an egg and you get a person who has cystic fibrosis and put the nucleus from the person with the condition into an egg and create an embryo from that. So it's basically cloning the person who has the disease and then looking at that cloned embryo as it develops and seeing how the disease arises. So there is another use that people have proposed for human eggs.
So another question, do you think that the embryonic stem cell research will lead to eugenic outcomes?
Well, embryonic stem cell research, so that embryonic stem cell research is making stem cells from human embryos. And if you want to make stem cells from human embryos, you have to have methods for allowing the embryo to develop to a certain stage before you dismantle it and turn it into stem cells. It may be that you can get better stem cells if you let the embryo develop a little bit further. So basically there's a drive when you are doing experiments on human embryos to see how far you can go, to see what benefits you can get. It may be that gestating this human embryo for 5 months in a volunteer woman's uterus will give you an organism, a human fetus that will be a great source of stem cells. So basically, if you embark on research using human embryos, and it's a kind of instrumentally based research, the objective isn't to produce a person, but the objective is to produce spare parts, for example, then you have a kind of incentive to create as near to a human as possible, but maybe not quite a human. And you also will develop the technology that will allow you to make improvements, if there's such a thing, by putting in new genes. So basically, embryo research facilitates both eugenics and what I've called myogenics. Myogenics meaning making worse versions of people rather than better versions of people, so that you can use them to help the health of other people.
So just for clarification's sake, so then they would be then essentially like you would part out a car.
Yes. Yes. Yeah.
Just checking. All right. So this takes us back to the beginning of the conversation, and the question is, what would happen if you would have actually gotten your patent? What would you have done next? And then there's two follow-up questions. Would that facilitate similar patents? And then would you have a duty to license it? License the technology?
Well, there are some legal scholars and some patent analysts that do talk about the obligation to license something that has been awarded as a patent. I think it's ambiguous whether there is really a legal legal impetus to do that. So, but I wouldn't have done it in any case. I wouldn't have licensed it, and wouldn't have. I would have raised barriers to anybody using it. I would have, if I were able to get the funding, I would have sued whoever wanted to use it, despite not being licensed. So I would have used the awarding of the patent to block the technology. Yeah.
Great. So let me see if I can get this right. So it says, comment on the tension between 2 points. The first point, if it is possible to do, sooner or later someone would try. And the second point is, but no one would really make a full-grown, chimera, right? And so then the question is, are we beholden to everyone's goodwill so that two will prevail?
Well, yeah, it's very difficult in the current environment to prohibit people from doing this in their garage. Well, some people have very well-equipped garages, so couldn't do it in most people's garages, but in some people's garages you could do it. People can try, and some people might be successful. In order to bring an embryo to term, you also need a woman who's willing to lend out her uterus for the period of time of gestation. It's interesting that there's a synthetic biologist named George Church who's just written a book where he proposes eugenic applications of synthetic biology, and he was interviewed in Europe about recent research on the Neanderthal genome, and he was looking forward to resuscitating Neanderthals as a species by enlisting women to gestate eggs that have Neanderthal genes. So there are strange people out there.
So I think the extinction stuff has actually been hitting the paper quite a bit, and I had an exchange with a good friend of mine who was at a conference over at Stanford where they were talking about that. I believe it was last Friday. And so is it technically feasible, or is this more sort of greasing of the skids? Maybe it's a stealth marketing for something that may emerge later.
Right.
Where do you?
Yeah, I think the second is more like, I think it's but it's an ideal that's held up. It's the idea about resurrecting Neanderthals or woolly mammoths or saber-toothed tigers. The idea is that if we can figure out what their genomes were, we could synthesize the genome, and we could put those genes in an egg, and the egg of a related species that's an existing species, and get these extinct animals back. Well, first of all, they won't be exactly the extinct animals because the egg, not only does it have its mitochondrial genome, but it has many factors that were placed in the egg by the mother during a process of oogenesis, which is the production of the egg. And these include regulators of the genes. And the regulators of the genes are not going to be exactly the same in a related species as they were in the original species. So the genes are not autonomous and putting out instructions to make something, and even though that is a popular idea that's very incorrect, but the genes are interacting with these factors, and there's this, as Elton John calls it, the circle of life, and and basically you get a new organism by the factors that are laid down during oogenesis interacting with the genes characteristic of that species. So you're never going to get the original type back. And even if you were to get one or two individuals, a species has an ecological niche. It lives in nature in interaction with other organisms, and this can never be restored. I mean, this is gone for these extinct species. So you might have a couple of zoo animals that are curiosities because they look like a saber-toothed tiger, but it's just it's just a very misguided venture.
It does, quite frankly. I mean, I could imagine somebody being initially sort of intrigued by it, but if you sort of think about it for a bit, it does appear to be a pretty problematic idea. I mean, I have to say, I was listening to a little bit of a videotape today, and somebody was talking about the passenger pigeon, and one of their rationales for bringing it back is it was a great game species. And so I'm thinking to myself, is that the reason why?
To kill it.
Yeah, let's bring it back. We can kill it and we can make some more. So a little more than a little
Monty
problematic.
Python routine about that.
At least, right. So here's a question that I actually think speaks to something I think would be of interest to a great deal of folks. So what has your experience been like trying to raise these issues within the scientific community? I know that sometimes when you push against sort of orthodoxy, you can become sort of persona non grata, or you're stifling somebody's, good ideas or initiative.
Right.
What have your experiences been like?
Well, I think there's a disincentive for scientists to do it because everybody is subject to other scientists approving their grant applications or their papers. So nobody wants to get on anybody's bad side, particularly if they don't have a stake in the scientific issues themselves. I don't know. I've actually found that I get a lot of agreement when I speak to people informally. I don't think I've ever met. I've never met a developmental biologist that's in favor of genetic engineering of humans. But I've rarely met developmental biologists that go on record as saying that they. So I think writing about these things, speaking about these things, I think it kind of creates a kind of a culture of caution. At least it helps to do that. So I think it's a worthwhile thing, but I don't see it creating a groundswell of opposition.
Great. So sort of along related lines, is genetic engineering in part just a general restlessness to fulfill professional roles, etc., to build on research or just to make contributions? Or is it just a justification for the expense and time taken for people to get their education?
In other words, why are people studying genetic engineering? Well, in my own field, a lot of deepened understanding of development has been made by genetically engineering mice, and it's a research technique, and a lot of synthetic biology are interesting research techniques. It allows you to manipulate biology in a way that allows you to understand biology better. There's the old ideal of pure science. I mean, these are very deep questions, how organisms develop. We're very far from understanding it, but we continue to make progress in understanding it. The problem is when people kind of oversell it, and somehow you can't. I mean, you can't be a pure scientist anymore, and look good to the funding agencies. People want to know what the implications for society are going to be. I mean, pure science has positive implications for society in that it contributes to the store of knowledge, and maybe down the line somebody can use it for something useful. But that's very different from this kind of upfront ambition of, I'm going to genetically engineer plants and solve a food problem, or I'm going to genetically engineer humans and make everybody smarter. It just that's just overselling, but it's more and more fashionable.
Great. So we're getting down to the last few questions, and some of them more or less run together, and some of them I think we've touched on, but just for the sake of it, I'll actually read them all out and see if we can sort of pull them all together. Two of them speak very specifically. Are folks doing this or planning to do this at Berkeley or at Cal? I think you touched on this a little bit, but isn't there a ban on human cloning in the U.S.? Yeah, so that's basically
Okay.
it.
Well, there are people doing synthetic biology and genetic engineering at Cal. They're not doing it on humans as far as I know. Some of them might be giving newspaper interviews that look forward to doing it on humans, but nobody's actually been approved to do it on humans in the United States. And there's no ban on cloning. There's a ban on cloning humans using federal money, but there would be no ban on somebody getting private money and attempting to clone a human being.
So that about wraps up the questions. I don't know if there's any remaining in the audience that we may try to field. Yeah. Professor Duster, please.
Yeah, Stuart, I want to tell my favorite chimeric story. It's about Stanford University. Several years ago, researchers there proposed to study the brain, issues of Parkinson's and Alzheimer's, by doing a project where they would insert millions of human brain cells into the brains of a very young mouse or mice. The committee approved this research, and the chair of the committee said there was no problem here. There's no issue, and I happened to be on a panel with him a very short time later. I asked him, well, what do you mean? He said, well, if there's a real problem, if these mice act more like humans, we'll just kill them. That's a quote. Okay? So I said to him, I said, Hank, how would you know when a mouse starts acting like a human?
Maybe you would prefer a different kind of cheese.
If that mouse was human enough to ask for his Second Amendment rights, it might not let it go.
And just to follow up on that, in that same series of experiments, one of the things that actually made it into the National Academies of Sciences guidelines was that one of the things that researchers should do is make sure that the personnel who were caring for the mice in the laboratory were trained, because in case they these mice started exhibiting any human characteristics, they didn't want the caretakers to freak out. That's in the NAS guidelines.
That's pretty out there. So it looks like we've about wrapped up. First of all, Stuart, I want to thank you for a lovely
Thank you.
I'm going to hand it back over to Tina, but I want to encourage folks to pick up the flyers outside. The series will resume again at the start of the next school year, very, very soon to that. And again, it's going to be an ongoing exchange. So thanks for coming out tonight, spending some of your time and energy. And I'm going to hand you back to Tina, and she'll take us home.
Thank you both very much. Very stimulating. So I told you that I asked you to hang on to these to the fact sheet about egg donation and the bill that's coming out on AB 926, and this letter. And that's because the Senate Health Committee is going to be considering this bill next Wednesday. And essentially what the bill is doing is it's trying to make it okay to pay women for their eggs for research. And we didn't really have too much time to go into what goes on, but it's really a very health risky thing for women to do. Women have been donating eggs for other women who are infertile for a few decades now. And despite the fact that many women's health groups have been asking the industry to do long-term follow-up on what the health risks are for women who donate eggs, they haven't really done it. And so we know that there are that women who undergo egg harvesting sometimes get ovarian hyperstimulation syndrome. You try saying that. Which can be very risky and leads sometimes, in rare cases, to death. But also, importantly, the long-term effects are not known. And in order to get women's eggs, it's not like getting sperm. You know, take this cup and come back in 10 minutes. It's actually it's a surgical process, but also it requires a lot of hormonal stimulation. You take very powerful hormones first to shut down the ovaries, put women in temporary menopause, and then you take very powerful hormones to hyperstimulate the ovaries to produce many more times the number of eggs than women produce a month. And there are at least 10 studies that show that there are concerns about long-term cancer effects. So it seems very injudicious, at least, to be asking women to undertake this for research when the risks have not been sorted out. So what we're asking you to do then is to sign this letter saying that you are asking them to not do that, to not pass this bill. And if you leave it with us tonight, if you didn't get a copy of the letter, you can get a copy at the door, or see Joan, who's at the back there. She'll come down and she can give you another letter. Just sign it, and we'll take it to Sacramento for you when we talk to the Senate Health Committee next week. And then the only other thing to do is to ask you to please come back, as Milton said, in September for our next East Bay conversation, which is Vandana Shiva, who will be talking about seeding labs, seeding seeds. Well, I guess the pun doesn't come out unless you look at it. Seeding is spelled with a C. And so the regional and global impacts of the biotech bonanza. I don't think everyone is aware that the East Bay has been dubbed the Green Corridor, which is where a lot of biotech labs are being seeded. And there's also been, recently there are several biohackers, just entrepreneurial people, who want to synthetically engineer seeds. And if you give them $40, they'll give you a handful of these seeds, and you can plant them in your garden or whatever. And they're supposedly going to glow green. And they're doing this. They started a Kickstarter campaign to get them in to do this. And of course, it's all without regulation. It's quite problematic. So if you go to the SynBioWatch website, which I hope you all do, you can see the Kickstopper campaign. And you can sign on to that. So please stay tuned. If you haven't signed up for SynBioWatch newsletter, please do that. And we'll see you next time in September. Thank you so much for coming.
Thank you. I enjoyed it.